This method can help answer key questions in the immunology field, such as the TCR alpha and beta repertoire of an antigen specific population, as well as the in vivo functionality of human TCRs. The main advantages of this technique are the relatively high efficiency of the PCR reaction, as well as its compatibility with subsequent TCR cloning. This protocol uses a two-step PCR to amplify the TCR alpha and beta chains from a single cell without prior knowledge of the TCR sequence. The first PCR is a multiplex reaction using a pool of alpha or beta variable region specific primers in conjunction with a primer specific for the constant region. The second PCR is a nested reaction that primes off of the adapter sequence incorporated at the 5' end of the PCR product, as well as an internal constant region sequence. These primers also incorporate unique restriction sites into the 5' and 3' ends of the PCR product to facilitate subsequent cloning. Once cloned into the template vector, the encoded TCR sequence is chimeric, consisting of human variable regions with murine constant regions. The chimeric TCR construct allows for interaction between the TCR and the murine CD3 signaling complex, making this TCR cloning protocol compatible with subsequent functional studies in murine cell lines and in humanized mouse models. Set up for the single-cell sorting by preparing the reverse transcription reagents in a clean, template-free area. Generally, individuals new to the single-cell PCR method will struggle because of the low levels of template. On the other hand, the sensitivity of the multi